Hi everyone, and welcome to our first topic for BIOL 112. We're going to look at vision. Now vision is a complex topic. There's a lot going on inside your eyeball. So I've decided to divide this into two parts. The first PowerPoint, part A, is going to look at the anatomy of the eye. So we're going to talk about the structures of the eye. The second PowerPoint, which is going to be a bit longer, is going to look at the physiology of the eye. So in part B, we'll talk about photoreceptors, what they are, how they're triggered by photons, and how they relay that information, that input, onto your brain. Now think back to Biology 111, and I know it was a little while ago. Try to remember what we talked about. We left off looking at special senses. Before that, we looked at general senses, and before that, we looked at the nervous system. So let's start with the nervous system. Remember that nerve cells are highly specialized cells that conduct electrical potentials. And that conduction of electrical potential is known as an action potential. So what's happening in a nerve cell is they are manipulating the concentration of ions that are found on the inside of the membrane and the outside of the membrane. And that differential in ion concentration is going to sweep down the length of the cell. There are sensory cells that can also produce action potentials. So a sensory cell is going to respond to an outside input. And that outside input could be temperature, pressure, a whole bunch of things. But that's going to create an action potential that will sweep down the cell and can excite another neuron, or could excite an effector, something that's going to bring about a change. So they might excite a muscle cell or a gland or something like that. Usually it's not direct. Usually you have a sensory cell exciting a nerve cell, maybe exciting a bunch more nerve cells, and eventually exciting a muscle or gland cell or similar cell. Now, in terms of sensory cells, we have two broad categories. We have general senses and we have specialized senses. And the name just refers to whether or not we're dealing with a specialized organ that houses those sensory cells. So let's look at the skin. The skin is a huge surface area, and it contains a lot of general sensory cells. So for instance, the skin will sense pain, the skin will sense pressure, vibration, temperature. Those are all general senses because they don't require a specialized organ. Now if we look at the special senses are vision, that we're going to talk about, and the three that we talked about last term, hearing, taste, and olfaction, or smell. So all of those four senses require specialized organs. We have the nose for smell. We have the tongue for taste. We have the inner ear for hearing. And we have the eyeball for vision. So you have general sensory receptors distributed throughout your body. You have special sensory receptors that are found within special sense organs within the head. You might be surprised to learn that 70% of all sensory receptors are found in the eye. And relatively speaking, the eye is a fairly small structure. I mean, compare it to the surface area of the skin. So why are there so many sensory receptors? It's because your eyes need to collect photons of light and build high definition, high resolution images of your surroundings. And that takes quite a lot of receptors, as we'll see. The eye is well protected. It's found within the orbit. Remember that the orbit is composed of bone. There's several bones of the skull that contribute to the orbit. And there's also a cushion of fat between the bones of the orbit and the eye itself. If we have a chance, hopefully you'll be dissecting eyeballs, probably horse eyes, that's what we usually do. And you'll see that there's a big wad of fat at the back. Accessory structures are structures that help support the eye, help lubricate it, or help move it around. So these structures would include eyebrows. The eyebrows are going to help reduce glare into the eye and also help keep out some dust and debris and other things that might damage the eye. You have eyelids that, of course, you can close, but also they sweep across the eye constantly to remove dirt and other debris. We have the conjunctiva, which is the lining of the eyelids, and it's also continuous with the visible white part of the eye. And this is a layer that produces a slippery lubricating fluid and also produces some protective elements as well. We'll come back to that. We have the lacrimal apparatus, which is the structures that produce and distribute tears onto the surface of the eye. And then we have the extrinsic eye muscles. Those are the muscles that attach to the outer part of the eye, to the sclera, which is the back part of the white of the eye. The part of the eye you should never be able to see unless something has gone terribly wrong. Let's start by taking a look at the accessory structures of the eye. And we'll start with the eyelids, or if you want to sound really fancy at a cocktail party, the palpebrae. So the eyelids or palpebrae are lined by the conjunctiva. And the conjunctiva is a mucosal membrane that produces a slippery lubricating fluid so that the eyelids can slide across the eye. The conjunctiva is also continuous with the whites of the eye. Now, you've probably heard of conjunctivitis before. The term itis, or the ending itis, refers to an inflammation. Itis means an inflammation of whatever preceded the itis. So conjunctivitis is an inflammation of the conjunctiva. Inflammation refers to when you have increased blood flow to an area. So the whites of the eye, which are lined by conjunctiva, are going to become red because of increased blood flow to the conjunctiva. So if we take a look at the diagram on the right here, and I'm going to use my little marker here to mark this out for you. Note that in order to see me drawing stuff on a slide, you will have to be looking at the slideshow and not just playing the audio. But we've got conjunctiva here on the inside of the eyelid, and that is continuous with the white of the eye here. So we have this continuous mucosal layer that is making the eyeball and the inside of the eyelid nice and slippery. Now, a few other things to note here. We'll come back to these later, but you can see some of the extrinsic muscles of the eye. So for instance here, wow, that's a bad arrow, but hopefully you get the idea. I'm trying to point up there. You can see lots of fat. So we've got a whole lot of fat in here, for instance, helping to protect the eye. You can see the eyelashes. The eyelashes will help keep dust and debris out of the eye. The lacrimal apparatus produces tears. You produce excess tears when, of course, you're in extreme emotional distress, and this is a sign to others that, you need some help. But you produce tears all the time, and those tears serve a rather important function. First of all, they lubricate the eye, so it's not going to be just stuck in your head, otherwise you wouldn't be able to move it around. But also, those tears contain a lot of important substances. They contain mucus, which helps with this lubrication. They contain antibodies, which, as you probably know, will bind to foreign proteins and tell your immune system that this is something that shouldn't be there. They also contain lysozyme. Lysozyme is an enzyme that specifically attacks peptidoglycan, and peptidoglycan is a mixture of protein and sugar that is specific to bacteria. It's only found within the cell wall of bacteria. So the tears contain a number of additions that will help fight against infection. I should mention that lacrimate in Latin means to cry. So it makes perfect sense that this lacrimal apparatus produces tears. And if you remember back to Bio 111 when you studied the skull, there is a little bone within the bony orbit that's known as the lacrimal bone. And the lacrimal apparatus sits on top of that bone. So we've got this fluid, tears, that are going to be secreted onto the eye, and the gland that does this is located on the upper outside corner of the eye, so the superior lateral corner of the eye. And in this diagram at the bottom here, you can see the lacrimal gland down here. I'll just circle it for you. Now, it's not usually that big. Hopefully yours is not that big. This is from a person that has an infection of the lacrimal gland. But even if yours is not infected, if you're careful, you can roll up your eyelid and you can see the location of that gland. So tears are going to be secreted by that gland, and then they're going to flow across the eye, and they're going to be collected in the medial corner of the eye, so towards the nose. From there, they're going to drain into something called the nasolacrimal duct, and that ultimately leads into the nose. Okay, so here's the flow of those fluids. We have the lacrimal gland, which is towards the outside upper corner of the eye, so it's lateral and superior. And then we have these excretory ducts that dump onto the eye. The fluid's going to flow across the eye and be collected towards the medial end of the eye. The fluids are going to pass into the lacrimal sac and then pass down this nasolacrimal duct into the nose. There's a few steps there that I'm not too concerned about, that you don't really need to know. But if you know the lacrimal gland, lacrimal sac, nasolacrimal duct, you're golden. I would say that's pretty good. Those are the major components of the lacrimal apparatus. Now, the tears flow into the nasal cavity. Now, once they get into the nasal cavity, the inside of the nose is lined by ciliated epithelium. So there's epithelial cells that have cilia that are constantly beating, and they're moving mucus and fluid back up into the nasopharynx and down into the throat so you can swallow it. So you're constantly swallowing a whole lot of crap from your nose, like it or not. It's pretty remarkable how much material you swallow from the nasopharynx every day. Now, what if it's a really, really cold day? If it's a really cold day, you're breathing in cold air into your nose, and that cold air actually chills the cells inside the nose. It actually limits their metabolic rate. It lowers their activity, and the cilia beat less frequently, and they can't actually move the fluid the way they would normally, and that's why your nose runs on a cold day. So the inside of the nose becomes chilled, those cells that should be moving fluid back are overwhelmed, and the fluid drips out your nose. Likewise, if you're crying, if something terrible has happened and you're under a lot of distress, you're producing excess fluid, and that excess fluid is moving towards the nose, and it's overwhelming the ciliated cells of the nasal epithelium, and it drips out of your nose. So we have this continuous flow of fluid across the eye. We have fluid being produced in the lateral corners of the eye, and then that fluid is moving across towards the medial corners of the eye, and then being shunted down into the nasal cavity, back into the nasopharynx, and swallowed. And that's happening all the time to wash the eye, lubricate the eye, and protect the eye. We also have eyelids, like I mentioned, that will sweep across the eye and help remove debris. And in a lot of vertebrates, a lot of animals, we have what's called a third eyelid, which is kind of what it sounds like. So we have, of course, two eyelids, an upper and a lower, but a lot of animals have a third eyelid that goes across the eye. And you're seeing an example of that here in a bird that does quite a bit of diving. Beavers, lots of aquatic animals have this third eyelid that moves across, and it's clear, and they can actually swim underwater with this eyelid closed to help protect their eye. But even animals that don't do a lot of diving have these third eyelids. If you want to be kind of mean to your dog or your cat and you just kind of poke them slightly while they're sleeping and they might open their outer eyelids, you'll see this third eyelid or nictitating membrane. Humans don't have this, but we have a remnant of it. So in the corner of the eye, and you can see it on that last slide that I showed you, there is a little fold known as the semilunar fold, and that's a remnant of this third eyelid. And incidentally, this is one of the things that I probably won't ever ask you about, but I just think it's kind of cool. Continuing with accessory structures, we have the extrinsic muscles of the eye. Again, X means outside. Think of X meaning exterior. These are muscles that attach to the outside of the eye and move the eye around. They're what allows you to look around without moving your head. And it may seem kind of scary at first, but the names actually make perfect sense. If you think back to the anatomical terms that you learned at the very beginning of Biology 111, we have superior, lateral, medial, inferior. Remember, superior means up, inferior means down, lateral means towards the outside, away from the midline, and medial means towards the midline, towards the center of the body. Rectus just means straight. So there's lots of muscles that are called so-and-so rectus. Rectus just means it's a long, straight muscle. So we have the lateral rectus, which is towards the outside of the eye. We have the medial rectus, which is towards the inside of the eye. And you can probably work out what they do. So the lateral rectus, if it contracts, if it shortens, it's going to move the eye towards the outside. It's going to make you look away from your nose, basically. Medial rectus, if it contracts, it's going to move the eye inward, and it's going to make you look at your nose. If you contract the medial rectus muscle on both sides, on both eyeballs, you're going to go cross-eyed. The superior rectus is going to make you look up. The inferior rectus is going to make you look down. So those make perfect sense. Now we have a couple that are kind of strange. We have the superior oblique, and we also have the inferior oblique. So superior again means up, inferior means down. Oblique means just that it's coming across at a weird angle. It's coming across at an oblique angle across the eye. Now, one thing to note, if we look on this image on the left, we have something called a trochlea up here that I've just circled. A trochlea is a pulley, and what we've got is this muscle, the superior oblique, that comes across the eye like this, goes over this pulley, and then comes back this way. And what it's going to do, as you can see here, is it's going to rotate the eye a little bit. It's going to move it up a bit, and it's going to rotate it a bit. And then we have the inferior oblique, which does kind of the same thing, but we're at the bottom here. It's going to rotate the eye a bit, and it's going to pull it down a bit. So these are muscles that you should know. These are muscles that we're going to talk about in the lab as well when we get to the muscle section of the lab. These are small muscles, but they're muscles that you have very precise control over. And remember that there are 3 cranial nerves that innervate the extrinsic eye muscles. There's one really important one, the oculomotor nerve. Motor implies movement. Oculo means to do with the eye. So the names make sense, and I really like to stress this all the time. Pay attention to what the names say. I mean, they seem scary. They're scary Latin or Greek names, but they typically do make sense. So you have this oculomotor nerve, nerve number 3, cranial nerve number 3, that innervates 4 of the 6 extrinsic muscles. We have the abducens, which is a odd little nerve in that all it does is innervate the lateral rectus, and the trochlear nerve, again, an odd little nerve that just innervates the superior oblique, and it's called the trochlear because the superior oblique goes through that little pulley. So there's a little ligament that's shaped like a little loop that redirects the force that's created by the superior oblique. And this brings us to the intrinsic muscles of the eye. The intrinsic muscles are muscles that are part of the eye. They're not outside the eye. They're not attached to the exterior. They are inside the eye. They are in the interior of the eye. And we've got two muscles. We've got circular muscles and we've got radial muscles. Now, the muscles make up part of what's known as the iris. And over top of the muscles, we have a pigmented layer. That's the colored portion of the eye. So the iris is the colored portion of the eye that you see when you look into someone's eye. In the middle of the iris, there's a black spot, and that's actually an opening. So that is an opening that allows light to pass back to the retina. And that opening is known as the pupil. So in really bright light, what happens is the circular muscles, which are in the middle, are going to contract, and they're going to restrict the size of the pupil. So they're going to constrict the pupil. In dim light, what's going to happen is the radial muscles, which are on the outside and attached to the circular muscles, are going to contract, and they're going to pull that inner circle out, and they're going to increase the size of the pupil. And that's known as dilating the pupil. So just to review, once again, we've got the pupil at the middle of the eye. That's an actual physical opening. It's a hole in the iris. And then around that, we've got the circular muscle. And around that, we've got the radial muscle. And the radial and circular muscle together make up the iris, along with a thin pigmented layer over top of that. The oculomotor nerve, which is cranial nerve number 3. Remember, cranial nerves are nerves that come directly off of the brain. Very important nerve when it comes to the eye. It controls 4 of the 6 extrinsic eye muscles, but it also controls the functioning of the 2 muscles that are found within the eye, the 2 intrinsic muscles. So if you recall, the autonomic nervous system is made up of 2 parts. We have the parasympathetic system and we have the sympathetic system. Parasympathetic system controls kind of ongoing stuff that's always happening in the body. We're generally talking about processes that maintain homeostasis. This is the rest and digest system. And then the sympathetic system, sometimes called the fight or flight system, is a system that increases activity with the body, that excites the body for fight or flight responses typically. So the parasympathetic part of the oculomotor nerve stimulates the circular muscle, and it causes constriction of the pupil. The sympathetic portion of the oculomotor nerve innervates the radial muscles and causes dilation of the pupils. And that's why if someone is very, very excited, their eyes tend to dilate, their pupils tend to get larger. And in fact, I'm told that let's say you're in a bar or something and you're flirting with someone. If they are listening intently to a story you're telling and their pupils are very large, means they're kind of excited. It means they're kind of interested in what you're talking about. Now, if you are in a fight or flight response, your eyes do tend to become very, very wide. And if you're in a very stressful event, they may stay in that condition for a while. So there are a number of drugs, for instance, that will cause the pupils to dilate. But if you come across someone who's been in a car accident, and they've been in this very stressful event, their body is going to be inundated with a lot of sympathetic responses And you're probably aware of this, but if you shine a light in someone's eye and their pupils remain very large, they don't constrict, it means that their eyes are being overstimulated by the sympathetic system. And it means that, this is a condition that should be dealt with. This person is undergoing a shock response. And this is what this looks like. So the person on the left has very, very wide pupils. This is something that might have been photographed under very dim light with a flash. So they've taken a picture before the eye has time to respond. Whereas the person on the right has very constricted pupils. So again, constricted pupil usually means you're exposed to bright light. Dilated pupil usually means that you're in very dim light situations, but it might also be representative of a very intense sympathetic stimulation. So now that we've looked at the accessory structures of the eye, and we've looked at the muscles of the eye, let's take a look at the structure of the eyeball itself. So the eyeball is almost a sphere, but not quite. It's a bit irregular, largely in part due to the fact that the cornea, the front clear part of the eye, sticks out a little bit. It's comprised of three layers, three different functional layers known as tunics. So think of a tunic as an item of clothing. It's a layer that surrounds something. We've got the fibrous tunic, which is the outermost layer. And as the name suggests, it consists of some rather tough fibers that are structural and help support the shape of the eye. Underneath that, we've got the vascular layer, which contains blood vessels, very fine capillaries and so on, that will help nourish the eye and provide oxygen to the eye and remove waste and carbon dioxide. And then the innermost layer is known as the sensory layer, although most people just refer to it as the retina. This is the layer that contains the photoreceptors. We have fluid inside the eye. So the internal cavity of the eye is filled with a fluid, and fluids found within the body have traditionally and classically been referred to as humors. This is an old Latin term for body fluids. The lens is going to separate the cavity within the eye into two parts, and those two parts are known as segments. So we have an anterior segment, which is in front of the lens, so it's in between the lens and the cornea, and then we have a posterior segment, which is the majority of that cavity. And taking a look at the diagram on the right, you can see the fibrous tunic consists of two parts. They're continuous. We have the sclera, which is the white of the eye, and then we have the cornea, which is just the front portion that's clear, the part that you can see through. We have the vascular tunic, which consists of a few parts. We have the iris, we have the ciliary body, which contains muscles that move the lens, change the shape of the lens. And then we have what's known as the choroid, which is going to provide nourishment and oxygen to the other layers throughout the eye. And then finally, we have the innermost layer. So that would be the sensory layer, which, as I said, is usually referred to as the retina, which consists of a pigmented layer and then a neural layer. The pigmented layer is a dark layer that contains a lot of melanin that's going to absorb any stray photons that have already passed through sensory cells, and the neural layer contains the sensory cells themselves. Let's take a closer look at the interior structures of the eye. So starting with the diagram on the left, if we look at the anterior of the eye, we've got the cornea, and this is the clear portion of the fibrous tunic. It's connected to the sclera, which is the non-clear portion of the fibrous tunic. So that's the white of the eye. The sclera is very, very tough. It's composed of dense, irregular connective tissue. Underneath the cornea, we've got what's known as the anterior segment, and this is the entire cavity that is anterior to the lens. So basically everything that's a cavity between the lens and the cornea. Behind the lens, we've got the posterior segment, which contains this jelly-like vitreous humor. There's actually a canal that runs through the vitreous humor, and that's that tube that you see running from the lens to the back of the eye, but we won't get into those details here. Looking at other layers of the eye, we've got, again, the sclera, which is part of the fibrous tunic. We've got the choroid, which is the vascular tunic, and then we've got the retina, which comprises part of the neural tunic, the innermost layer. Note that the retina, shown in yellow here, is continuous with the optic nerve. Now, what's happening is within the retina, we've got photosensitive cells. So we've got photoreceptors that respond to photons of light. And from the retina, we're going to have axons that will leave the retina and travel back as the optic nerve back to the brain. The optic nerve is hollow. And you can see that we have a central artery and vein within that hollow of this nerve. It's an odd nerve in that it has a hollow canal that runs down the middle of it. And that has to do with how the eyeball develops, which we'll talk about briefly later on. At the back of the eye, we have something called the optic disc, also known as the blind spot. And this is where these arteries and veins enter and leave the eye, and in that area there are no photoreceptors. So in this area here, I'm just going to circle this. In this area here, there is no retina because we have that space. We have that cavity that contains this artery and vein. There are no cells that are capable of forming an image in that spot. Now up above this, we have a very interesting area here. It's a little off center. It's not directly at the back of the eye, but this is where most of your vision comes from. This small area here, known as the macula lutea, and the fovea centralis, which is in the middle of this region, is responsible for the vast majority of your high resolution or high acuity vision. And you're seeing a close-up of that area in the photo to the right. So the photo to the right was taken using an ophthalmoscope, which is that device that they shine into your eye, and the eye doctor will look through this and have a look at the back of your eye and look at the blood vessels of your eye. So let's take a look at that image. First of all, back here, I'm going to circle this in blue. This area here is the blind spot or optic disc, and this is where you can see there's blood vessels that are entering the eye. That area, once again, has no photoreceptors, and that's why it's known as the blind spot. You can't form an image on that small part of your eye. Now, the part that's already circled in green is the macula lutea, and the part that is outlined in black is the fovea centralis. And this is a fairly small portion of the eye. So in this image here, we're actually zoomed in quite a bit on the back of the retina. Now the macula lutea and the fovea centralis, like I said, are responsible for most of the high resolution vision that you get through the cones of your eye, which we'll talk about in the next PowerPoint. The fovea centralis is actually a small depression, and within the fovea centralis we have nothing but cones, and cones are responsible for high resolution color imagery. Okay, let's just take a look at these different tunics, these different functional layers in a bit more detail. So once again, we have the fibrous tunic. It's the outermost layer. It's a supportive layer, so it's structural. We have the opaque sclera towards the posterior of the eye, and this is the white of the eye. We have the cornea, which is the anterior most part, which is clear. It has to be clear, of course, to let light through the pupil and ultimately back to the retina. The sclera is going to protect the eye. It's going to maintain the shape of the eye, and the interior of the eye is actually under a little bit of pressure. So the fluid within the eye is exerting a little bit of outward force, and the sclera is resisting that. The sclera is also where the extrinsic muscles attach, so the muscles that move the eye. The vascular tunic or choroid region contains the blood vessels that help nourish the eye. This also includes the ciliary body and the iris. So we've got this kind of dark brown membrane that forms the posterior part of the vascular tunic. That's the choroid. That's the majority of the vascular tunic. Lots and lots of very fine blood vessels in there. And it's going to provide nourishment and oxygen to the other layers of the eye. The ciliary body is part of the vascular tunic. It's this thickened ring of tissue that is in close association with the lens. It's connected to the lens through these suspensory ligaments. And as I mentioned, there's smooth muscle that is going to pull on the lenses and change the shape of the lens. So it can pull on the lens and cause the lens to flatten or to constrict, which is going to change the focal point of the lens. The iris, again, still part of the vascular tunic. It's the colored part of the eye. So in this image here, it's that blue part. And right under that pigmented layer, we would have the circular and radial muscles of the eye. So the iris is going to control the size of the pupil. And again, the pupil is just an opening. It's just the hole. If you're in a situation where there's very bright light, the pupils are going to constrict, but that also happens during close vision. So if you're reading something up close, your pupils will constrict. For dim light situations, the pupils are going to dilate to let in more light, and the pupils will also dilate when you're looking at something that's very, very far away. Emotional state can affect the size of the pupil. We talked about already how the autonomic nervous system can affect the size of the pupil. So pupils tend to dilate if you're very interested in something, if the subject matter is appealing to you or requires problem-solving skills, or if you're just experiencing a sympathetic nervous response. So if something is interesting and you're gathering more information about it, your pupils are going to open up to gather more light. Similarly, if you're in a stressed situation, if you're in a fight or flight response, your pupils tend to dilate. The sensory tunic, generally known as the retina. So the retina consists of two parts. We have what's called the pigmented layer, and this is a layer that absorbs light and prevents it from scattering. So this is an outermost layer. And you can see in this diagram here, it's that very, very thin black layer. Above that, we have the neuronal cells. So we have photoreceptive cells, and we have some other cells as well, known as bipolar cells and ganglion cells. And we'll get into that in a little bit. But what's interesting is light that comes into the eye has to pass through all these nerve cells. Not all of them are directly sensory, but the photons have to pass through that layer of nerve cells, and then any photons that have not triggered a response are going to be absorbed by this pigmented layer, because otherwise the photons would bounce around inside the eye and trigger a number of different photoreceptors, and you wouldn't get a nice, clear, high-resolution image. There's other cells known as amacrine and horizontal cells that are going to regulate the response of the retina. We won't get into those in too much detail. It's actually quite fascinating the way the retina works. It's a really complex little bit of machinery, and it's not fully understood. We're taking a closer look at the back of the eye here. You can see the optic disc there. That's where this canal in the center of the optic nerve enters the eye. And that canal contains the central artery and vein. Again, the optic disc or blind spot doesn't contain any photoreceptors, and that means we can't form an image in that small area. The yellow strands that you see leaving and forming the optic nerve are coming from cells known as ganglion cells. They're the axons of ganglion cells. Ganglion cells are not photoreceptive cells, but they connect to them indirectly, as we'll see. So they ultimately receive signals from the photoreceptive cells, the rods and cones of the retina, and then those ganglion cells are going to transmit a signal through axons. The axons are all bundled together and they leave the eye as this hollow optic nerve. The optic nerve is not involved in moving the eye. That's a mistake a lot of students make when they're studying the cranial nerve. So the optic nerve is cranial nerve 2, and all it does is receive sensory input from the eye. It's not involved in innervating the intrinsic or extrinsic muscles of the eye. It just receives sensory information from the eye. Let's just take a closer look at the retina itself. So once again, we've got a diagram here to the left that shows the optic disc or blind spot. Again, no receptors there, as you can see. And within the retina itself, we've got our photoreceptive cells, which are shown in this layer. So we've got rods and cones. And then underneath that, we have this pigmented layer down here. And the pigmented layer is going to trap any photons that make it all the way to the back of the retina without being captured. The photoreceptive cells are going to send off a signal if they receive a photon. Now that's not quite accurate. I'll come back to that later on, but for the time being, that's a suitable explanation. They're going to send a signal through this layer of cells to these ganglion cells here. And the ganglion cells contain axons, which are thread-like extensions of the cell that can conduct an action potential. They're going to conduct an action potential through their axons all the way back to the occipital lobe of the brain. And the optic nerve is just a big bundle of axons from these ganglion cells, with, again, a hollow in the middle that contains the central retinal artery and vein. And once again, you've got a view through the ophthalmoscope there showing the macula luteae and the fovea centralis, or just fovea. This is the area where we have the largest concentration of photoreceptive cells. Let's take a quick look at photoreceptors. Photoreceptors are sensory cells that respond to photons of light. Now, we're not going to spend too much time on this right now because this is something we'll talk about quite a bit in the next PowerPoint. But we have two types. We have rods and we have cones. Rods are more numerous, but they're more sensitive. So rods respond to very dim light. In fact, it's thought that one photon can activate a single rod, which is pretty amazing. They're found throughout the eye, but especially the peripheral area of the retina. So the periphery of the retina has only rods and no cones. They're very sensitive, as I mentioned, and that makes them very, very good for perceiving peripheral movement. So if something moves on the periphery of your field of view, you typically recognize that immediately, which makes sense. It could be a threat, could be something to eat. So if we go back to our hunter-gatherer days, having a good sense of when things enter your periphery was quite a good skill to have. Cones, on the other hand, not nearly as numerous, and they require many photons to function, but they respond to different wavelengths of light. So cones are responsible for color vision. Rods just give us a grayscale or black and white field of vision. So if you go out on a dark night, you have a hard time seeing colors. You can just see shadows and shapes and so on. Cones provide us with high acuity, which is a fancy term for high resolution vision. And the cones are found almost entirely within the macula lutea. Within the macula lutea, remember, we have a pit in the center, a very small pit, known as the fovea. And the fovea is entirely cones. So why are the cones focused here? Well, it's because the lens focuses light on the macula lutea, and specifically the center part of that, the fovea centralis. So the majority of your high acuity color vision is actually coming from a very small area of the retina. Now, the one last thing I think is kind of interesting here is the pathway that light takes through these layers. So notice in the diagram here, the bottom right would be the interior of the eye. That would be the posterior segment. Light is coming through the posterior segment and then passing through the axons of the ganglion cells, that's shown in yellow. Those axons are going to come together and form the optic nerve. So all of these axons here will form the optic nerve that leaves the eye. The photons of light, after they go through this layer of axons, have to go through the ganglion cells themselves, and then through the bipolar cells, and then to the photoreceptors. And even then, the photons may not get absorbed by a photoreceptor. If they don't, then they hit the pigmented layer of the retina. And this contains the pigment melanin, which is really quite effective at absorbing photons. The reason for that is to prevent any stray photons that didn't get absorbed from bouncing back into the inside of the eye and activating other rods and cones and giving us a fairly blurry image. Now, there are some animals that see well in the dark that do have a reflective layer at the back, and that reflects photons back towards the cones and rods, and that does give them a better chance of collecting photons and forming an image, although it is at a lack of acuity. So if you think of cats, for instance, also cows, for instance, if you shine a light into their eye at night, you see that light bouncing back to you, and that's because of the reflective layer. That's something we'll talk about in lab if we have a chance to dissect cow eyes or pig eyes or whatever. Now, there's a few other cell types that I'll mention very, very briefly. The rods and cones are going to ultimately activate ganglion cells, and there's an intermediate in between, and that's the bipolar cell. Now, notice that there's also what are known as horizontal cells and amacrine cells, and these are going to regulate the activities of nearby ganglion cells, in the case of the amacrine cells, and nearby bipolar cells, and also rod and cone cells in the case of the horizontal cells. And what this does is basically it turns one area of the retina into a pixel, if you want to think of it like you'd think of a television screen. But we'll come back to that in our next section. Very briefly, I'll talk about the blood supply to the retina. Photoreceptive cells, like other nerve cells, are very, very energetic cells. They need a lot of oxygen, they need a lot of nourishment. So we have lots of blood vessels that supply the eye. The one we've talked about so far is the central retinal artery that travels through a canal in the center of the optic nerve, and that goes into the eye and carries blood to the retina. But we also have blood vessels that radiate off and surround the eye and supply the lacrimal gland, supply the muscles that move the eye, and some of them actually punch through the sclera and supply the choroid with additional blood. The chambers of the eye and the way they're named can be a little bit confusing. We have two broad cavities. The posterior segment, which is the majority of the interior of the eye, so this is the cavity behind the lens. It contains the vitreous humor, which forms something called the vitreous body, which is just a blob of this rather dense, gelatin-like vitreous humor. In front of the lens, we have what's called the anterior segment. Now, where it gets confusing is the anterior segment is divided into two cavities itself. So we have the space between the lens and the iris, and that's known as the posterior chamber of the anterior segment. And then in front of the iris, we have what's called the anterior chamber of the anterior segment. And that can be kind of confusing. And if you're looking at different sources online and so on, you'll find that some people do misuse these terms, but I'm going to use the terms as they're presented in this diagram here. The anterior chamber contains aqueous humor. Aqueous means water-like. So the fluid within the anterior segment is fluid, much more fluid than what we see in the vitreous body, which is quite gelatin-like. The posterior segment contains fluid that's under pressure. So that vitreous body, that vitreous fluid is under pressure and it's pushing out against the outermost tunic, the fibrous tunic. Think of the eye as kind of a basketball. So you have air under pressure in a basketball. Basketball itself is quite elastic. It's pushing back, and that helps maintain the structure of the basketball. In much the same way, the fluids in the eye are under pressure. They're pushing out against the fibrous tunic. Fibrous tunic is quite elastic. It's pushing back, and that helps maintain the structure of the eye. There are several disorders where we can have a disruption in that balance between push and pull, between pressure within the eye and pushing back of the fibrous tunic. So if the circulation of fluids within the eye is disrupted, we can have a buildup of excess pressure within the eye, and that can be rather painful. So once again, we have the posterior segment, and then we have the anterior segment, which is divided into two chambers, the anterior chamber and the posterior chamber. Aqueous humor is plasma-like. Plasma is the fluid portion of the blood, and basically that fluid portion of blood leaks out through blood vessels and forms the interstitial fluid between cells and forms the aqueous humor in this case. So fluid, excess fluid that leaks out from the blood is going to replenish the aqueous humor within the anterior segment. And what it's going to do is it's going to provide oxygen, it's going to provide metabolites like glucose to cells that need it, and that's also going to wash over the cells and remove waste products. I don't expect you to know a whole lot of detail when it comes to this, but we have capillaries within the ciliary body of the vascular tunic that are leaky. So blood is going to leak out plasma, it's going to leak out fluid, and that fluid is going to flow out into the anterior chamber of the anterior segment, and we have this circulation basically that occurs. That fluid is going to be collected into what's called a venous sinus that leads into venules that lead into veins, and that gets back into the general circulation of the circulatory system. Main thing to note here though is that if we have a problem where we're not producing enough fluid, then you could have a situation where the eye isn't fully inflated, for lack of a better word. That's not that common, but we can get blockage of that venous sinus, and that means that fluid is being released into the eye, and that fluid is not being removed from the eye. And that means that the eye is going to swell. And as I mentioned before, that can cause some issues and some pain, as you might imagine. The lens is the structure in the eye that focuses light on the retina at the back of the eye. It's transparent, of course, so that light can pass through it. It's biconvex, which just means that it's curved on the front and the back. It's curved outwardly. It's flexible, and in fact, it's kind of elastic. So if it's not being pulled on by the suspensory ligaments, it tends to take a rounder shape. If it's pulled on by the suspensory ligaments, it flattens out. It's avascular. Vascular means that there's blood vessels. Whenever you see A at the beginning of a word, that generally means without. It's kind of a Latin convention. If you stick an A on the beginning of a term, it means without. So avascular means that the lens doesn't have any blood vessels inside of it. It allows for light to be focused on the retina, and we'll talk about that more in the next PowerPoint. It's composed of epithelium and also specialized lens fibers. Again, we'll talk about that a bit more next class. The lens fibers are filled with a transparent protein called crystalline, and this forms crystal-like structures within the lens. As we age, the lens become less flexible, they become less elastic, and we unfortunately become less able to change the shape of the lens. And that's why it's very, very common for people to require glasses, especially for reading as they age. I'm going to end this PowerPoint with just a very brief discussion of the development of the eye and also the optic nerve coming out of the eye. So what we've got in the upper left here is a very early human embryo. There's little pouches here that make up part of the pharynx, which develops into the throat. And then above that, we've got an eye forming. And we're looking at a section of the eye up at the top here. And you can see there's a big hollow space in the middle, and that is the brain. So the brain is a hollow organ. Even when you're an adult, there are hollows within the brain known as ventricles. At this stage of development, the brain and the spinal cord are just hollow tubes. And we have these little pockets that form here that bud off of that hollow tube, and they stretch out, as you can see. So that's that hollow tube here. They stretch out and they form the eye. And they form a little channel. So we can see this channel through here, and that's the channel that persists within the optic nerve. So the optic nerve is unusual compared to other cranial nerves in that it has a space in the middle. And the reason for that is because the cranial nerve and the eye itself are basically little lobes of the brain. So they're little offshoots of the brain. Now it's not shown in these other diagrams, unfortunately, but that canal continues back to the brain. It's quite a narrow little canal, and it's just filled with those little blood vessels. The eye itself forms from a little cup. So you can see that this little lobe that comes off of the brain forms a cup, and then we have this little ball that forms within that cup that forms the lens. And then the very last step that happens here is that we have a layer on the outside of the eye, and this is continuous with the skin, that forms the cornea, forms the outer part of the eye. So I'm going to leave things there for now. As I mentioned, this is a big topic, so we're going to continue it on in the next PowerPoint. At the end of the next PowerPoint, I will have a list of terms that I expect you to know. So I don't expect you to know every little detail that I talked about, but I will give you a list of terms, and those are the terms that you should focus on. Next class, I'll also be posting a study guide that will outline some of the major points for today's lecture and next day's lecture. Okay, so hopefully that was a nice start to the term, and we'll continue this next day.